inside the cells, how the replication actually takes place. So the last class we saw the basic mechanism by which all the proteins that is involved in the replication. So let's say that type of replication it's a little bit different in different organisms. So in prokaryotes, it's a little bit different because it's just a circular genome, a small genome. And in eukaryotes, it's a very large genome, polyploids, many number of chromosomes. So how that replication can take place in the prokaryotes, the bacteria, the phages, and also the eukaryotes, right? So starting with bacterial bacterial chromosome replication. So all that synthesis, the DNA synthesis begins at the origin of replication. We have already seen this. There is an ORI. ORI is origin of replication. So each bacterial chromosome has only one origin of replication. So this synthesis of DNA proceeds bidirectionally around the bacterial chromosomes. So that's a linear chromosome or the DNA. So that DNA goes bidirectionally on both the sides, right? So the replication forks eventually meet at the opposite end because this is a circular bacterial chromosome, is a circular DNA. So that the replication proceeds bidirectionally. So that we gave an example of the cycle tire where you have the tube and the tire just separating from each other, right? So that ends, the end of the replication starts when the replication fork meet at the opposite side of the bacterial chromosome, there is the end of the replication. Right. So starting with the origin of replication in the bacterial chromosomes. So the replication fork proceeds bidirectionally. So the one is this way and the other one is this way. So that these two DNA separates and this ends where this replication fork meets. So you have a separate protein. So protein here that stops the replication so that it doesn't go beyond. So it will not be a rolling circle in the sense. So it will not be just be rolling around, right? So it starts at one end, it proceeds at one direction, and the other one is the other direction, so that you have a copy of this. And when these two replication forks meet, so here around, so you will have a separate protein that stops the replication, and these two DNA separates together. The chromosome separates together. So the initiation of replication in bacteria. So the origin of replication in E. coli is termed as this ORI-C, the origin C. So origin of chromosomal replication. So this mostly consists of the DNA sequences. There are three types of DNA sequences. So these are called as this consensus sequence, right? We will see this later on. Consensus is it's the same sequence that is present in almost all organisms. So this sequence will be recognized by the proteins, the replication proteins. Say like consensus means the same sequence present in all the organisms, so that it is easier for the proteins like this the DNA-C, the ORI-C proteins to recognize and bind to it. All right. So it's a consensus sequence. Three types of DNA sequences in the ORI-C are functionally significant. So one is the AT-rich region. So there will be a small stretch of region consisting of 80 regions. Then the other one is the DNA A boxes. So where the DNA A protein comes and binds. Right, so this is a DNA A box, and there is a GAT-C methylation site. So this site, so there will be this three sites in the sequence where this origin of C is replication, ORI-C is present. The ORI-C is just these three consensus sequences, right? So this in that ORI-C in the E. coli chromosomes. So this stretch, like starting with around this 275 bases. So you all this DNA sequence will be starting from the 5' to the 3' end, and it will be numbered. So starting with the nucleotide 1 to nucleotide 50. So this ORI-C region consists of around 275 bases. So where you have this AT rich region, TA, 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 TA region, right? AT rich region, and you have the DNA A box. DNA again a consensus sequence TGT, TA, TGT, TA, TGT, TA, TGT, TA, all this boxes, right? So this type of consensus, so this type of sequence is common in all the organisms, right? So these type of sequences will be recognized by this DNA A protein, right? So in this origin or REC region, you have this AT rich region, which will be recognized by the other proteins, replication proteins, and you have the DNA A boxes, right? So separated throughout this length of the DNA, right? So you understand from, so until here you understand, right? So coming later on, we will be seeing many of those proteins. So you might not need to remember all the proteins, but this is the type of replication initiation in almost all the organisms, right? So எப்படி நடக்குதுன்னு மாத்திரம் நம்ம தெரிஞ்சுக்கலாம் அது, not remembering all those proteins, because eventually we will forget all the proteins, and we are still it is being researched how the replication occurs, right? So first, so there is some region that will be bound by a protein, and the other DNA A boxes, and the sequences which will be recognized by the DNA A protein, right? So this, the DNA replication, which is initiated by the binding of DNA A protein to the DNA A box sequence. Like some protein called the DNA A protein comes and binds to this sequence, right? So they read through those sequences, the proteins. So that just reads through all the length, and when it recognizes this type of consensus sequence, it will go and bind to that, right? So this, starting with this binding of this protein stimulates the cooperative binding of additional 20 to 40 DNA A proteins to form a large complex. So once one DNA protein recognizes this sequence, so that will bring in all another 20 to 40 DNA complexes, and then all this bind to this region. So this, the other proteins like the HU or the integration host factor also bind to this AT rich region. So you don't need to remember all this. So many types of proteins comes and bind to this, right? So this causes the regionate to wrap around this DNA A region and separates the AT rich region. So what happens is, so when this HU, the host integration factor, so comes and binds to this AT rich region. So this separates this AT rich region, the initiation region. So now there is a replication 4 forming, right? So not the helicase. So this is also an helicase that separates and forms the replication 4. So this DNA A protein as a single protein along with the other 20 to 40 DNA A proteins like bends onto this, all those consensus sequences, right? So this, the DNA B protein is also a helicase which separates the DNA bonds, the hydrogen bonds between these two sequences, and it forms the replication fork, right? Starting. So the DNA C, also another protein, assist in this process, but later on it is released. So this, which is also in helicase DNA B, which is also in helicase, it will start breaking the hydrogen bonds and separates the two DNA strands, so which eventually forms the replication fork. So this helicase composed of 6 subunits, it travels along the DNA in the 5' to 3' direction, and it uses the energy from ATP, and this helicase separates the DNA in both the direction, creating 2 replication fork, one here and the other one on this side. So when this breaks the bonds and then keeps moving, the polymerase also binds here and synthesizes DNA from the 5' to the 3' end, the other one from the 5' to 3' end. Yeah. So there is a bidirectional replication of this DNA in. So this is what we are seeing is in bacteria, E. coli, right? So the DNA helicase separates the 2 strands by breaking the hydrogen bonds between them, and this generates the positive supercoiling ahead of each replication fork, and DNA gyrase, or also called as the 2' isomerase, right? So this travels ahead of the helicase and alleviates this breaks this supercoils. And also the single strand binding proteins bind to the separated DNA strands to keep them apart. And then the short 10 to 12 nucleotides RNA primers are synthesized by primase, by primase enzyme, and the DNA synthesis starts. And this primers are later removed and replaced with the DNA. Right? So this primase and the removal will be called as Okazaki fragments. Right? You remember this. So you start this helicase, which opens up this double bond, and ahead of this, there is this topoisomerase releasing the tension in the back behind. And this DNA polymerase synthesizes from the leading strand, right, where this single strand SSB, single strand binding protein, keeps the two strands attaching together. And in the lagging strand, you have this RNA primer that is synthesized by this primase enzyme, and the Okazaki fragments are synthesized from this RNA primer in the 5' to 3' direction in the DNA in the lagging strand. Right? It's the same. This picture is the same what we saw earlier. Right? So breaks the hydrogen bond between the two strands. The helicase and topoisomerase alleviates the supercoiling, and the SSBs keep the parental strands apart, and the primase synthesizes the RNA primer. So the DNA polymerases in the bacteria, these are the enzymes that catalyze the attachment of nucleotides to make the new DNA. And there are 5 proteins in the polymerase activity. So the first DNA pole 1, 2, 3, 4, and 5. So this is in E. coli, right? So there are 5 different polymerases. So the DNA pollen 2, which is pollen 1 and 3. This is required for the normal replication, and 2, 4, and 5. So these are required for the repair and replication of the damaged RNA. Right? So each unit of the DNA polymerase 3, the holoenzyme. So DNA polymerase 3, which synthesizes the nucleotides, so consists of those 10 different subunits. So we don't need to remember all these things. So again, the polymerase is also made up of different subunits. Right? So that the alpha subunit synthesizes the DNA, this epsilon proofreads theta, this stimulates the proofreading function, or these are all accessories proteins. Accessories protein bind to the single strand binding protein. So each of those components bind and has a different function in this DNA polymerase 3. This is the DNA polymerase 3, which synthesizes the DNA. So the DNA polymerase 1 removes the RNA primers and fills the gap in the DNA. So it uses the 5' to 3' exonuclease activity to digest the RNA, and its 5' to 3' polymerase activity to replace it with the DNA. So after the gap is filled, a covalent bond is still missing in those attachments between those two different newly synthesized one, which is catalyzed by the DNA ligase, which joins using the phosphodiester bonds, thereby connecting the DNA fragments. So coming to the end of replication, so opposing to the RHC is the pair of termination sequences. So there is also a termination sequence which is called as the TER sequence. This is in, again, bacteria. So do not forget that all this is in bacteria, not in eukaryotes. So these are designated T1 and T2, terminator sequence 1 and T2. So the protein TUS or termination utilization substance, the TUS protein binds to this sequence and stops the replication. So this it can then stop the movement of the replication forks. So like when the opposite ends meet together. So this termination 1 sequence here. So this will not allow forwarding in the clockwise direction. And this, the termination sequence 2, so this is in the opposite direction, right? So this will not allow in the counterclockwise direction, and TER1 will not allow in the clockwise direction, right? So this will stop here. The replication will stop here. So when the fork emerges, so the TER binding or a termination binding protein attaches here, and it will not allow the anti-clockwise. And here, when it attaches here, it will not allow. So this does not allow clockwise, and this will not allow anti-clockwise. So allows the advancement of the clockwise moving fork, and the other one prevent the movement of counterclockwise moving fork. So T2 allows the advancements of counterclockwise moving forward, and the other one is the opposite, prevents the clockwise moving forward, right? So this, when this is present here, this will not allow the clockwise. And when this is present here, this will not allow the counterclockwise, right? So this termination of replication, so the DNA replication ends when the opposingly advancing the forks meet usually the T1 and the T2 regions. So finally the DNA ligase covalently links all the 4 DNA strands. So where there are gaps, so all the DNA ligase closes the gaps, and the DNA replication results in 2 intertwined molecules, right? So still, both the molecules or the 2 different chromosomes are attached, so which is called as the catenins, right? So when both are attached, when these are not separated, so these are separated by the action of the topoisomerases by breaking the single or double bonds, right? Again, topoisomerases not only just releases the stress, also releases the replication, the replicated 2 catenins. Say like for example here, so you have the DNA, the chromosome, so which replicates into 2 different. So these are still attached, right? Those are still attached. So this is called as the catenins, right? So there can be several. So it can, when it replicates, so there can be 2, like 2 rings together, attached together, right? So these 2 rings, so there can be many rings also. So when this 2 rings are attached together, these 2 rings are called as the catenins, right? Catenin is a chain, right? So decatenation by this topoisomerases just by breaking a single bond or a double bond, it releases the 2 different chromosomes. So catalysis by the DNA topoisomerases. So the DNA replication complexes, the proteins that are involved, the DNA helicase and primase. So these are physically bound to each other, which is called as the primosome, right? So the helicase and the primase, which adds the primer, DNA primer, and also the helicase. So these 2 together is called as the primosome, and both the proteins are called as the primosomes. So this complex leads the way at the replication fork. And the primosome is physically associated with the DNA polymerase holoenzyme, forming the repliosome. So the primosome first creates the replication fork and moves. And when the polymerase, the DNA polymerase comes and attaches to this primosome, it will be called as the replisome. So starting with this, so first the helicase and the primase. So first helicase open the DNA this way, right? So forming the replication forks. So the helicase and also the DNA primer, which adds the primer. So this both together, helicase and primase, both together is called as the primosome. And when this is also attached by the, helicase and primase, when this DNA polymerase also attaches to this primosome, it is called as repliosome. All the 3 proteins together is called as the repliosome, right? So this is the actual picture. So where in a bacteria, when it replicates, so you see this 2. So this is the actual chromosomes be replicated into 2 different catenins, right? So this what is called as the theta structure, right? So this is the theta. Like this forms a theta structure, which suggests that the replication started in only one place on this chromosomes. So the replication of circular DNA and the supercoiling. So there are 4 important points when this replication takes place. So the first, there will be a supercoiling problem. So when this topoisomerase releases the supercoiling, or the helicase starts to form this 2 different replication forks, so there will be supercoiling behind this chromosomes. So this in the circular chromosomes, there are circular throughout the replication and creating a theta shape. As the strands separate from one side of the circle, positive supercoils form in the back end of the molecules. So the replication fork moves about around 500 nucleotides per second. So the replication takes about around 500 nucleotides per second. So at 10 bases per turn, the replication fork rotates at around 3000 RPM. Right, so the tension, whatever the behind the replication fork, so the DNA rotates at around 3000 RPM, creating a supercoil. So this supercoil is released by the topoisomerases, right, allowing the DNA strands to continue separating as the replication fork advances. So this would be the theta structure. So when this two molecules, the catenanes, are replicating together, so there will be this formation of the supercoils behind where this is being replicated. And each time when it progresses, so the topoisomerases breaks either the single strand or both the double strands and releases all the supercoils behind this. So this is in bacteria, and the next one is in phages, right? So or in the viruses. So the example of the adenovirus. So it is almost the same. It's also in the viruses, or in the phages. So the DNA polymerase is the same, having the 5' to 3' polymerase activity, and all known DNA replication system all require this free 3' hydroxyl group before the synthesis can be initiated. Right? So this four different mechanisms of initiation of synthesis is present in the viruses. So the viruses, phages, plasmids use a primase to synthesize a short RNA primer with a free 3' OH group, which is elongated by the DNA polymerase. And the retro elements, including the retroviruses, employ a transfer RNA that primes DNA replication by providing a free 3' OH that is used for elongation by the reverse transcriptase. So it's the same. So it is in different wordings. So what happens is, again, in all this DNA viruses, so this is in RNA viruses. So where you have this primase, first starts the DNA synthesis, which provides the free 3' OH group. So starting with this. Okay, so here, so most of this in the viral DNA is circular DNA. So what you don't see here in this picture is that so starting with that with the transcription the termination protein, that TP is the termination protein. So this adds the 3' OH group to the single strand. So you have this double strand here. So this protein comes and binds to this, where this termination protein attaches and gives with this single strand binding protein. So this is the SS double strand, single strand binding protein, which attaches to the single strand and starts synthesizing, or the primers to this. So all this, so when there is a strand displacement, or when the two strands are separated, so there will be synthesis of these two different strands. So in the first strand, after the first strand is completed, so this strand will form again another template, right? So this template strand will again be recycled, and this will start synthesizing again. So after all the synthesis, so this DNA, so all this, most of this bacteria, I mean the phages and also the retroviruses, you have a single strand. Some can have single strand DNA or RNA. So that can form circularization, and it forms a circular DNA. So what happens here is that in adenoviruses and also in the theta 25 family of phages, the 3' OH group is provided by the side chain of an amino acid of a genome attached protein, which is called as the terminal protein. So this terminal protein comes and attaches to the single strand, to which the nucleotides are added by the DNA polymerases, which form the new strand. Right. So the adenoviruses, there is a precursor terminal protein, the PTP, which forms a complex, and the DNA polymerase serves as the primers for the DNA replication. Right. So again, coming back to this, all this replication processes, because this DNA viruses or RNA viruses, it is all that we need to separate it, right? Instead of forming this replication fork in this DNA, where you have the single strand DNA or a single strand RNA in the viruses. So you have this terminal protein coming and attaching to this, which gives the 3' OH group, free 3' OH group, and forms a primer, where the DNA polymerases adds the nucleotides to this. Right. So after adding the nucleotides, those single strand circularizes, and this forms the genome. So there will be again after forming this, there can also be again formation of the second strands, and this can replicate. Replication can go on and recycling. So this is in the viruses. So in the single strand DNA viruses and the group that includes the circoviruses, Gemini virus, paravirus, and others, and many phages and plasmids, uses the rolling circle replication mechanism. And this endonucleases creates a nicks in the genome in the single-stranded viruses, or one of the strands of the DNA in the plasmids, and the DNA polymerase synthesizes the new strand. So mostly whatever we have seen in the viruses is the rolling circle replication, which is done by this endonucleases and the DNA polymerases. So in the DNA replication in the phages, the replication is the rolling circle mechanism, which is used by the bacteriophages, including PyX174 and also lambda after circularization between the pairing between the sticky single-stranded cos ends. So this rolling circle replication begins with a nick or the single-stranded break at the origin of replication, and the 5' end is displaced from the strand, and the 3' end acts as the primer for the DNA polymerase 3, which synthesizes and continuous strand from the intact DNA molecule as the template. So it is the same whatever we are seeing here, except that in here you have this rolling circle mechanism, right? Not as straight like whatever we have seen in bacteria. So the 5' end continues to be displaced as the rolling circle rolls, and it is protected by the single strand binding protein until discontinuous DNA synthesis makes it a double stranded DNA again. So this circular DNA, replication of the circular DNA molecule in that rolling circle mechanism. So nick is made here. So wherever you have this origin of replication, so a nick is made, and this strand is separated by the single strand binding protein, and this single strand binding protein attaches to the single strand, and the DNA synthesis takes place from the 5' to the 3' direction, which is called as the repliosome. And this actually keeps on rotating, and then in the other strand there's an old Okazaki fragment which also makes up the another strand of DNA. So this goes on rotating and rotating, forming catenates and then releasing the DNA. So the next last one is the DNA replication in eukaryotes. So it is the same. So whatever we have seen in bacteria or the phages, it is the same in eukaryotes also, except that we have several of those chromosomes, not a single chromosome, just like bacteria or viruses. There are many different chromosomes. It's similar to both the prokaryotes and the eukaryotes, except that the eukaryotes have more than one chromosomes. And this eukaryotic chromosome contain multiple origins of replications, multiple origins at which the DNA denatures, and the replication then proceeds bidirectionally until the adjacent replication fork is encountered and stops. So the DNA replication form a single origin, which is called as the replicon, or also called as the replication unit. So there can be many replication units or the replicon in each chromosome of the eukaryotes. So the DNA replication from multiple origins of replication. So each chromosomes. So you have the centromere and also the telomere, where we can have many number of origins of replications in the chromosomes. So during the S phase, the before S phase, the chromosome is intact, and during the S phase, during the synthesis of the DNA, so there can be many replication forks in each of those origins of replication. Right, so this replication starts bidirectionally, and each of those lengths where the origin of replication starts, so there will be replication here. So there will be a bidirectional DNA synthesis. So the DNA synthesis extends to both ends until it reaches where the two replication fork meets here. Right, there will also be there will be a termination sequence where this needs to release. That replication fork needs to dissociate. So the replication forks will then merge together and stop and merge together, and giving two separate sister chromatids. Right, when this two separates, this forms the two separate chromatids, where this is the signaling the end of S phase of replication. So this will be the actual picture. So where we can have multiple replication units in a single chromosomes. So you can have multiple replication units in each of those chromosomes, which then extends and then join together. So there can be multiple origins of replication found in eukaryotes, and which have with some similarities to those of bacteria. And in yeast, yeast it is called as the ARS, or autonomously replicating sequence. Right. So those origins of replication, it is called REC in bacterial chromosomes, and in eukaryotes it's called as ARS, or autonomously replicating sequence. So these are all 100 to 150 bases in length, and they have again the same requirements. It is AT rich region, right? High percentage of ATG, and have 3 or 4 copies of a specific sequence which is similar to the bacterial DNA A boxes. So the initiation of replication. So this first, the initiation is recognized by what is called as the CDKs. CDKs, or cyclin dependent kinases. So these are all the proteins. So in bacteria, you have this DNA A, right? DNA A protein that binds to the REC. So in eukaryotes, it will be the cyclin dependent kinase, or CDKs, that comes and binds to this origin of replication, or the ARS in the eukaryotes. So these are present throughout the cell cycle except during the G1 phase. So in absence of CDKs during the G1, the replication selection occurs, and the RC and other proteins assemble on each of this replication to form this replication complexes. And when the cell enters this S phase, the CDKs are present and activate the pre-replication complex to initiate the replication. So the CDK activity inhibits another round of pre-replication complex formation until the cell again enters the G1 phase when the CDKs are absent. So what this means is, so in the absence of CDK, first there are the replicative complexes are formed. So after the complexes are formed, the CDKs comes and attaches and then takes up the replication, initiation of replication. So after both this replication forks come in contact to each other, so this CDK activity inhibits another round. So before again going in another round of replication. So this again the cell enters the again G1 phase, and then the CDKs will also be absent then, and then this cycle again goes on. So starting with here, so this will be the G1 phase, which will be the start. So when the CDKs is present, so the G1 cyclin is present and inhibit the action of the CDKs. So when it starts to the S phase, so this G1 cyclin is degraded, and this will start the synthesis of DNA, or the replication in the S phase. So all of the protein, the mitotic cyclins or CDK, CD2 cyclins, all this start to synthesize. And when this again comes back to the G1, this cyclin comes and binds to the CDK, and it will not allow the again replication of the another round of DNA. Right? So basically what you need to remember is the previous diagram, right? Formation of the replication fork, and then there are few of those the presence of the sequence DNA boxes like AT rich regions are essential for the binding of the DNA A proteins and the other polymerases, right? So the enzymes, so it's the same almost in all bacteria, phages, and also in the eukaryotes, except the few proteins are involved or not involved. So this replication can be in 5-mark question, right? So this eukaryotic replication enzymes of eukaryotic DNA replication. So these are all still not characterized. We still don't know what are all the other proteins that is involved and how it is being replicated. It is still being researched. So the replication process to similar in both prokaryotes and also in the eukaryotes, where the DNA denatures. The replication is semi-conservative and also semi-discontinuous, and primers are required. So almost all these 4 requirements are present in both prokaryotes and the eukaryotes. Right? First, DNA denaturation of DNA by the helicases. Then the replication is semi-conservative and semi-discontinuous. Right? So there can be several of those bubbles forming and also it needs primers for the synthesis of the DNA. So in mammalian cells, there are 15 different DNA polymerases. So each, the 3 DNA polymerases are used to replicate the nuclear DNA. Polymerase alpha extend 10 nucleotides RNA primer by about 30 nucleotides, pol delta, pol epsilon. So this extend the DNA RNA primers, one on the leading strand and the other on the lagging strand. The other DNA polymerases replicate the mitochondria or chloroplast DNA are also used in DNA repair, right? So there are again many polymerases that is present in eukaryotes. You don't need to remember all those 15, 16. Still, we are again researching and finding out there can be many other polymerases, right? So this again, the polymerase types, it has several different functions. So alpha, theta, epsilon, gamma, all those subunits, different types of polymerases having different types of functions for DNA replication. So DNA polymerase alpha is the only polymerase to associate with the primase complex and synthesizes the short DNA RNA hybrid. So 10 RNA nucleotides followed by 20 to 30 DNA nucleotides are formed. And this is used by the DNA polymerase epsilon for a progressive elongation of the leading and also the lagging strand. And we still don't know, there might be a greater role for the DNA polymerase theta. Right? So still, there can be many other roles for this polymerases in the eukaryotes. So again, coming back to this, so you have this primase which adds the RNA primer and going from the lagging strand and also in the leading strand in a progressive manner. So that is done by the DNA polymerase theta and alpha are the primase synthesizes the RNA primer, and this proceeds in the bidirectionally in the eukaryotes also. So at the ends of chromosomes, so this is the important part. So in the synthesis enzymology, we have seen that after the RNA primer is degraded, so there will be no end for the synthesis of DNA, where you have the telomerase enzyme synthesizes a specific RNA primer, right? You remember that? So telomerase is the enzymes adds the RNA primer to the ends of chromosomes. So when the RNA primer, actual RNA primer is degraded, telomerase adds the primer to the ends of chromosomes, right? So if it does not add, then each time the chromosome replicates, then the chromosome will be shortened, right? So when the ends of chromosomes are replicated and the primers are removed from the 5' end, there will be no adjacent DNA strand to serve as the primer. So a single stranded region is left at the 5' end of the new strand. So if the gap is not added, the chromosome, so it becomes shorter with each round of replication. Right? So this, so when it starts replication, so there the RNA primer at the end of it will be degraded at the both the strands. Now strand here and also the strand here. So this RNA primer will be degraded and there will be a gap in both the chromosomes. So which when it is not added, so this single stranded will be eaten away by the endonucleases, exonucleases, and this will get shortened by the every cycle of replication. So this will be added by the telomerase enzymes. So each of those, so this is the synthesis of the telomeric DNA by the telomerase enzyme, right? So this, the gap left by the primer removal. So the RNA of the telomerase, so the telomerase has the RNA inside, which adds as the primer. And when this adds some primer to this, and then there will be a closure, adding of those nucleotides to these ends. So when this attaches to the nucleotides from the 5', the 3' to the 5' as the template and 5' to 3' as the extension. So from here, the polymerases adds the nucleotides in this way, 5' to the 3' end and closes the gap. Right, each time it closes the gap, and this will be ligated by the ligase, DNA ligases. Right, so this is actually, we can actually see how what the age of the chromosome is. So the termination, in the termination, the eukaryotic initiation of the DNA replicates at the multiple points in the chromosomes, and so when the replication fork meet and terminate at many points in the chromosomes, and these or not known to be regulated in any particular way. So in the bacterial and also in the phage, so we have seen that the TUS proteins. So that terminates the sequence. But here we don't know any of those termination sequence or any other proteins that is being involved. Right. So this actually stops the replication fork stops when it comes to the end of it. So because eukaryotes have the linear chromosomes, the DNA replication is unable to reach the end of the chromosomes, but the ends of the telomere region of the repetitive DNA close to the ends. So in each of those shortens the telomeres of the daughter DNA strand. So this shortening of the telomeres in the normal process in the somatic cells. So this can only divide certain number of times before the DNA loss prevents further division. So when the gap is not closed and that chromosome length decreases every time of replication, so that is called as the Hayflick limit, right? So Hayflick limit gives how many times the chromosome can divide, right, or replicate or make new copies. So when the telomeres length gets shortened, right? So you understand, we saw in the last class. So each and every time the chromosome replicates, right? So each and every time when this gap is not closed, right? So this length actually comes down. So in the telomeres, you have only the repetitive sequences, which is not of much use. But when this is not filled, then there will be not enough telomeres left, which will eat away into the genes present here. So how many times this lengthening, shortening of length can be done is called as the Hayflick limit of those chromosomes, right? So if there is excess, so there is one possibility that this needs to be closed and there will be shortening of the length of the telomeres. Or there can also be addition of more of telomeres here, right? There is also another possibility because presence of this telomerases can also add another sequences to the top of it. If it doesn't add, it will go down, right? That is called as the limit. Or it can go on adding more. So if it starts to adding more, there will be no death to the chromosomes. If there is no death to the chromosome, it will be called as cancer, right? So this is the in situ hybridization of the presence of this telomerases, right? So telomerase acts on the ends of the chromosomes where it adds to the single strand and then adds to the ends of the chromosomes, and each and every time when there is not enough addition of this, so if a single cell it divides, so you have this length of telomere. So every division, so you have this, so the first division you have this length, and the second division the length is reduced, and the length is reduced in the other division, and as it goes on there will be no more telomeres left. So this will be the Hayflick limit for any cell. So when any of those cells reaches this end, that will be the end of the cell, so that this cell will be recycled. So every time, even if it is our skin cell, it needs to be replenished. It should not be non-dyed cell, right? It should be replenished, and this is the Hayflick limit for each and every cell by which it can replicate, right? So when the germ cell line, which passes the DNA to the next generation, telomerase extends the repetitive sequences of the telomere region to prevent the degradation of the chromosomes. And the telomeres can also become overly active, mistakenly overly active in somatic cells, adding more of the telomere region and making the cell non-diable, right? Non-dead, sometimes leading to the cancer formation. So which increased telomerase activity is one of the measurements of formation of cancer. So the termination requires the progress of the DNA replication fork, which must be stopped or blocked, and the termination of the specific locus, when it occurs, involves interaction between two components, the termination site sequence in the DNA and also the protein which binds to the sequence to physically stop the DNA replication. And it can also be some TUS or TER protein can also be involved in this eukaryotic DNA replication. And after that DNA is replicated, and they are replicated, the immediately complexes with the histone proteins so that it does not get entangled after it has replicated, immediately it will be wound onto the histone proteins, so that these two again does not again intertangle together. So the transcription of the histone genes is initiated near the end of the G1 phase, and translation of histone protein occurs throughout the S phase, and assembly of the newly replicated DNA into nucleosomes. Right, immediately after replication of the single strand DNA into two data strands, immediately it will be enclosed by the histone proteins. So the newly made histone after this replication. So first the histones are released from this DNA, and after the two daughter strands are synthesized, immediately the new histone proteins form complexes with the new DNA, so that these two does not get entangled. So this is the replication process in the eukaryotes and the prokaryotes. Right. So this the replication process in prokaryotes can be in 5-mark question, which is almost the same in the eukaryotes. Right. So any other question? So all this there is an animation here. All right. So you can see how it can replicate here.